Hi everyone. Today I'll be talking on second MBBS exam pattern in microbiology according to the competency based medical education assessments. This exam, the theory will be given for will be conducted in two papers. One is paper 1 and paper 2. Each paper carries 100 marks. And paper 2 also carries 100 marks. In the syllabus, general microbiology, immunology, cardiovascular system, gastrointestinal tract and hepatobiliary system, skin soft tissues and musculoskeletal system. The questions will come from these chapters for the paper 1. And for paper 2, the central nervous system, respiratory system, and genitourinary system, and zoonotic diseases, oncogenic viral infections, and congenital diseases and other miscellaneous infections will be given in the paper 2. Pattern for this question paper pattern for this theory exams, as I mentioned already, the questions paper will come for 100 marks. One modified question is which carries 15 marks, and one case based or problem based where the case scenario will be given, that is for 15 marks, and 10 short notes will be given for 50 marks, and each carries 5 marks. And then among these 10, 2 are problem based questions where a short case scenario will be given, and one question related to the ADCOM, that's attitude, ethics and communications related related in healthcare system will be given. The question will be given from that aspect, and there are 20 MCQs. This each carries 1 mark, and among these 20 MCQs, 3 are case scenario with case scenario where the 1 or 2 lines of case scenario will be given, and 2 based on the reasoning based on the case scenario. Why you have to choose that particular option, there should be some reason, and this depends on that particular aspect, and 15 MCQs are direct questions. This is an example for case based or problem based question. This is related to the enteric fever. It's related to the enteric fever. So based on this particular case scenario, you have to come to the proper diagnosis. Unless you come to the proper diagnosis, then entire thing will go off. See, this is related to the enteric fever. No, this recently when I asked the same question in the question paper, some people wrote about the rickettsial infections. Some people wrote about rickettsial infections, and some people wrote about the brucellosis. Like this, if you go wrong, the entire thing, the questions asked, this kind of questions will be answered for this particular kind of diseases. So always you have to diagnose it properly, and you have to answer for their relevant questions given. Number 2 is a modified structured essay question, and here the essay question is with certain objectives. Here in this question, if you see, you have some objectives to answer. One is definition you have to write, and classification of hypersensitivity, and their immunological aspects you have to write, and pathogenesis you have to write, and clinical manifestations of this type 1 hypersensitivity you have to write, including its laboratory diagnosis, and each objective will be given some marks. See, 3, 4, 3 like this. So unless you answer in all the aspects of this particular type 1 hypersensitivity, you will lose marks. So while you are studying also, you have to study in this manner. What is the definition, and how to classify hypersensitivity reactions, and what is the mechanism of type 1 hypersensitivity, and how the disease will be produced, and what are all the clinical manifestations, and how to diagnose it in the by using laboratory test, all these things you have to write, otherwise you won't get full marks. Next problem is practical examination. Most of the times people will feel very much tense at the time of practical examination, and even though they know the answers and even though they are having the skill to perform certain exercises like staining procedures, they may not able to get good marks. So to avoid it, you have to follow certain guidelines while you are appearing for practical examinations. First of all, while you are coming for the practical examinations, you have to look good. That means your appearance has to be very good. You have to dress with neat dress, and you have to wear the shoes, and your hairstyle has to be appropriate, and you should have, out of all, you should have your ID card with your registration number, and clearly visible registration number has to be affixed either on the white coat, or you have to make your own arrangements, but it has to be visualized properly. Then you have to appear for one by one. One is the spotters for 10 marks. The 10 spotters will be given, each carries 1 mark, and gram staining for 15 marks, and special staining, maybe acid fast staining, maybe Jaden staining or quinone staining. There are two are there. It's either Jaden or quinone stain. You have to, you will be given either one of these, or Albert stain. This will be given Albert's. This is a special stain. These are the two important special stain you have. You will be given either quinone stain or the Albert stain. Then applied microbiology is another one, and the stool examination and the another exercise, and the objective structured practical exercise examination for 10 marks. Here I will explain you later. And AdCom, while you are in the hospital environment, while you are performing certain things, certain procedures on the patient or in the patient care, you may come across with certain untoward incidents. How to combat those incidents and what is the care to be taken by applying your attitude, ethics and communication skills will be asked here in the form of questionnaire. And then that carries 5 marks, and then you have marks for record and logbook. But remember, this is only for internal assessment. In the place of viva voce, you will be given the marks for your record and logbook. And viva voce in university examination, there are 20 marks for viva voce. There are 4 examiners will be there, and each one will award 4 marks, and total 20 marks will be given for the student. Spotters, as I mentioned already, 10 numbers will be given, and each carries 1 and 10 marks. Most important thing which you have to remember is, first you have to identify. First you have to identify the spotter, and once you identify, write it clearly and legibly. Write it clearly and legibly with proper spelling. This is most important, proper spelling. If there are any difficulty in spellings, you have to practice the spellings. And then don't forget to write 2 salient features, minimum 2 salient features of this particular spotter. Minimum 2 salient features, that means related to that particular thing. All this has to be done within 1 minute time, because the time will be given for this spotter is 1 minute only. So don't forget to write the name with proper spelling and 2 salient features. If you won't write the salient features, you will get only half mark. Don't expect 1 mark without writing the salient features of the spotter. This is always for 15 marks. And again, the 2 types of smears we will prepare and will be given for the students. One is marked with P and another is marked with E. So this P sample consists of the pus cells pus cells with staphylococci. With pus cells with staphylococci. Don't write the name of species, because based on this smear you can't say whether it is Staphylococcus aureus or Staphylococcus epidermidis or saprophyticus. So don't write the species name, just write pus cells with staphylococci. And also the diagram also should consist of only pus cells with staphylococci. And then emulsion. This emulsion is a mixture of gram positive cocci and gram negative bacilli. So you have to draw the diagram. While you are drawing the diagram, always you have to draw, in case of what we call pus, you have to draw the pus cells like, see, the cocci should be like this only, not like this. Some people will draw the diagrams like this. So should not draw like this, or very big circles like this. So these are all wrong. Don't draw like this and don't draw it, but you have to draw like this only. You have to draw very small. And then pus cell. The pus cell will be always the pink in color. Here the pink is not available, so I am taking red, and don't consider it as pink. So the pink colored, always the pus cells will be seen like this lobes. Here the pus cell, some people will write like this, draw like this, one drop, and this is not pus cell. This is not the pus cell. That means I will show here in bigger way. This is not the pus cell like this. This is wrong diagram. This is how we have to show the lobes. But this pus cell is degenerated polymorphs, so it should be like this only. The size of the pus cell should be like this, because this is around 10 to 15 microns in size, whereas this is around 1 micron in size. So the ratio should be maintained while you are drawing the diagram. So this is the diagram for bacteria, and this is diagram for pus cells. And then next important thing is you have to label. You have to label it properly. It's a diagram. Always this circle also you have to use either in the bangle or any wash bottles are available in the laboratory while you are doing this procedure. So use that wash bottle and then make a circle and then draw the diagram neatly and legibly, because it gives a good impression for the examiner while they are examining you. And the next one is, in case of mixture or emulsion, you have to draw the diagram. Again, this, see, in case of bacilli, you have to draw the bacilli like this, regular bacilli. The size of the bacilli is a regular size, should not be more than this. Again, the cocci, usually we will give again the mixture of staphylococci there. Then you have to draw the cocci like this only. So here and there, some groups you have to maintain. Here and there you have to maintain certain, like solid masses. Don't like circles, because the cytoplasm of the cell is being stained here. So only you have to show them as solid masses. So this is how you have to, and again you have to label. It's gram negative. You have to write like gram negative. Don't like G and B, gram negative bacilli. Completely you have to write. And this you have to write gram positive cocci. Don't mention that whether it is a staphylococci or something. You have to write like this. And while you are writing, don't write like this, gram positive and gram negative like this. Don't write like this. You have to write complete name, that is gram positive bacilli and gram positive cocci. Like this you have to answer, and the viva will be there. So always you have to read your record properly. Read your record. Read the record before coming. It's be very easy to write down the observation. Write the observation and inference. The inference. Don't mix up your observation with inference and inference with the observation. Read the record, and everything has been given there, and 1 or 2 lines of observation and a single line of inference is there. So you read and write it properly. And once again, pus smear is marked as P, and emulsion is marked as. So diagrams you should not get mixed the diagrams. If you are given with P, you have to draw this diagram, this particular diagram, and if you are given with E, you have to draw this kind of diagram, right? In this acid fast staining, again only one smear is available. It's marked as A. It is marked as A, marking as A, right? So this is prepared with sputum. This smear is prepared with sputum obtained from case of pulmonary tuberculosis. It's a case of pulmonary tuberculosis. So once a smear is prepared, then you have to stain it properly, and then the staining procedure. You don't write staining procedure on your answer script. Staining procedure you have to by heart. You have to by heart staining procedure. And the most important step in staining procedure is decolorization step. Of course, even gram staining also with decolorization only, and here also the decolorization step is more precious step. And the end point, this is not time bound. Decolorization step is not time bound. Not time bound. Don't tell that decolorization has to be done for 1 minute or 2 minutes like that. This end point for decolorization, is we have to decolorize the smear till the brown colored fluid ceases to come from the slide. So that is the end point for decolorization. Till that point you have to decolorize. It's not like 1 minute, 2 minutes, 3 minutes, something like that. If the smear is thicker one, it will take some more time. If it is a thinner one, it will take lesser time. So this is not time bound procedure. Decolorization step is most important. And here we use, previously people used to say the 20%, but according to the RNTCP guidelines, is a 25% H2SO4 has to be used as a decolorizing agent. 25% H2SO4 sulfuric acid has to be used as decolorizing agent. So after that you have to use 1% methylene blue. Methylene blue has to be as counter stain. So after that, once it gets over, you have to draw the diagram. Here the diagram, again here, always you have to carry the blue pencil. Don't forget to carry the blue pencil here. Blue pencil always have to carry. And so the pus cells, you have to draw the pus cells with this what we call blue pencil only, because you are using methylene blue as counter stain, no? So the pus cells have to be drawn with blue pencil. And again with this bacilli, as I am telling you that this is the red I am taking because the pink is not available in the screen. So here you have to draw the diagram like this. It's a long slender bacilli with occasional branching, here and there, occasional branching. So like this you have to draw, but don't draw the diagram like this much big and branching. And some people draw in the internal assessment like this, see, like this, and again like this. So these are all the wrong diagrams. These are all very big bacilli, and some people may draw like this with the beaded appearance, means they'll draw like this. This is all not correct. See, this is that beaded appearance is rarely seen, but this is the correct diagram. This is the correct diagram for drawing to draw at the time of examination, and it has to be labeled properly. And this has to be labeled properly, and this is also has to be labeled properly. So the pus cell with long slender bacilli with occasional branching. Because of that branching only they are called mycobacteria, right? So this diagram is very important. And then if you go for the inference, in the inference never forget to write acid fast bacilli Don't write like this. Just I am to cut short the time I am using this. Acid fast bacilli resembling Mycobacterium tuberculosis. Don't forget about this proper spelling. Here the important thing is why you have to use this resembling. This resembling word carries very important role because some of the saprophytic mycobacteria, will resemble the Mycobacterium tuberculosis. Just their morphology would be like that of Mycobacterium tuberculosis. That's why you have to write resembling. Where this saprophytic mycobacteria are present. This saprophytic mycobacteria are present in the environment. So while you are preparing the smear, there is a possibility for contamination, or while doing the staining, that is procedure, at the time of procedure, they may contaminate the smear and gives false positive results. So that's why please don't forget to write that word resembling. Always you should write the word resembling, acid fast bacilli resembling Mycobacterium tuberculosis. For the rest of the things, read, record again. I would like to say that read, record. All the questions and answers also have been given there, and all the questions related to that have been given there. Please read that one. So for this one, the pencils what I have to take, I have to carry are, one is blue pencil and another one is eosin pencil These two pencils are very important, and HB pencil for labeling. So these are all very important things to carry. Don't forget to bring the blue colored pencil. And so please ensure that your pencil is really giving this kind of violet color or not. Don't get mischief by, don't get into the troubles just by seeing the color of the pencil on this outside. Sometimes they looks like the wooden part looks like violet in color, but the lead part will give the blue color. So you check it whether it's blue or violet, and if you draw the violet in the place of blue or blue in the place of violet, again you will lose the marks. The representation should be proper. Aspect, applied microbiology. Here the examiner will test you whether you are able to apply your knowledge. Are you able to apply the knowledge for the diagnosis of the disease or not? So that is the purpose of this. So here the one case scenario will be there, the case scenario, and after this case scenario you will have the culture media, and also the biochemical reactions will be provided. Biochemical reactions will be provided, and like otherwise called the biochemical reactions used for the identification of those bacteria, all will be provided. So based on these findings, based on the findings on the culture media and the biochemical reactions, you have to identify the causative or identify the etiological agent. You have to identify. That is your final target for this particular exercise. So agent. So this is about the applied microbiology. If, say for example, if you get any case of wound infection, and based on case scenario, when the examiner asks you what is your case scenario, you have to tell this only. But the causative agent would be staphylococci. Don't tell that the staphylococcus aureus or something. You have to tell that what is your case scenario. You have to tell wound infection or enteric fever or pneumonia or some of the herpes virus or herpes labialis, like that you have to tell. So the case scenario is for the wound infection, but after culturing on media and by doing the biochemical reactions, we may identify it as Staphylococcus aureus. So for all these, the relevant questions are already given in your record. So go through your record, and read all the relevant questions. Have to read the record and the relevant questions, and the relevant questions and answers have been provided. So please read them, then it will be very, very easy. So there also very short 4 or 5 questions, not more than 4 or 5 questions have been given on that particular chart. And this, if you answer these 4 or 5 questions, that's more than sufficient for documentation. Then viva voce will be there. Then viva voce on that particular case. On say for example, if you get the Staphylococcus aureus, all the matter related to the Staphylococcus aureus will be asked in the viva related to this applied microbiology. Again, you have to the charts will be provided. The charts nowadays, the samples are not easy to get, so charts are being provided. Then you have to identify them. And while you are identifying, don't forget to put the number. The number will be written on the table nearer to the chart, so you have to put the number. And once you put the number, then identify. See, the parasites may be seen either in the form of trophozoites or cysts in case of protozoans. Right? But in case of helminths, you may be seen, they may be seen in the form of ova or larva. Again, this larva may be filariform larva or rhabditiform larva. So you have to mention that whether either it is filariform larva or rhabditiform larva, or you have to write ova or something, or parasite. So these are the 3 important forms of parasite, a single parasite in the helminths. So say for example, if we keep the ascaris, egg of ascaris, don't write it as ascaris lumbricoides. This is how they will write. But you have to write this, whether it is fertilized or unfertilized, also you have to mention. Say for example, fertilized egg of ascaris lumbricoides. This is the correct name or correct identification of egg of ascaris. Don't write like just ascaris lumbricoides. Even the same thing, trichuris trichura. In case of trichuris trichiora. So if usually we will keep the egg of trichuris trichura, then you have to write egg of trichuris trichura. Don't write just like that, trichuris trichura, egg of trichuris trichura. Same thing in case of entamoeba histolytica, you have to mention whether it is cyst or trophozoite of entamoeba histolytica we focused. Don't write like entamoeba histolytica. Don't write like this. So this is not correct answer. You have to mention whether it is either cyst or trophozoite. Okay. After that, you have to, after identification, what you have to do is, you have to draw the neat label diagram. So for this neat label diagram, for this you have to practice. All the diagrams. Practice all the 7 or 8 diagrams you have to practice. In classroom also I also mentioned what are all the eggs and what are the larvae. You have to practice and you have to draw in the examination. And again, they have to be labeled. Draw labeled diagram. These are the things you have to practice. Again, the relevant questionnaire will be there. pertaining to that particular egg or ova, ova or cyst or parasite. Structured practical exercise exams. There are three exercises will be given. Three exercises will be given, and two each carries 2.5 marks. 2.5 marks, total 5 marks. Here what we give is we provide you either culture media or instrument or some vaccines or viral models. Any one or any two will be given, either culture media, instrument, vaccine and viral models, just like what are all there for you in your department for keeping as spotters. The same thing will be given, but difference here is there are one or two or sometimes three questions will be given. Say for example, if we keep the blood agar medium and we will give three questions related to that. So you have to answer only those questions, but not at all the information what you know. You have to answer what the question related to that particular blood agar. So in that way you have to answer this question. Only this is a written part. There won't be any viva in this. Only written part. You have to identify, answer these questions, and you have to hand out to the examiner. You have to go. And one more is the skill, related to your skill. The skill, it carries 5 marks, and the what is that checklist will be there with the examiner, checklist, and according to the checklist you have to perform your skill. Say for example, hand washing is there. Hand washing, there are some steps are there, no, in hand washing. So if you perform each step, it will be some marks will be avoided. If you perform all steps properly as mentioned, you will get 5 marks. So here there is no documentation and no interaction with your examiner, just you have to perform. The examiner will award marks. Another thing is biomedical waste management. Again the white color, blue color, yellow color, red color and black colored, all these things will be there. Containers will be there. So all the biomedical waste, it's the simulation, it's only done in simulation, and simulated samples related to the biomedical waste will be provided, and you have to discard them. You have to discard them in the proper container. Discard in proper container. So this is also well explained in the practicals, so you can read your practical record. Ethics and communication related exercise. Here you have the needle stick injury, related to the needle stick injury, and one more is related to the bad news like hepatitis B viral infection or HIV infection or HCV infection or any kind of cancerous condition. So like this the case scenario will be given, and the relevant questions also will be provided. You have to answer them. Another thing is spill management. While the people are taking the blood or carrying the blood, there is a possibility for what we call the spillage of blood. So how to handle that spill will be given. So these are all related to the attitude of the individual and ethics to be followed, and the communication skills related to that particular healthcare personnel. So this exercise also will be provided. Your practical examination and how to do it in the perfect manner, especially in the staining procedures. You have to be very careful, and for all these things you have to read your record properly. My sincere advice to all you is, before going to the practical examination, just read your record. Just read your record. You need not to read your textbook at all. If you read record, this everything will be given there. It will be more useful for you even to attempt vivavos also. Okay. So all the best. Take care.